Today we are learn interesting topic PCR. Okay, PCR means polymerase chain reaction. Okay, this is very important technique. Okay, okay. PCR means P means polymerase, C means chain, R means reaction. Okay, polymerase chain reaction. Okay, PCR is a laboratory. Okay, laboratory technique means in vitro technique, okay, for generating large quantities of specified DNA. Okay, obviously PCR is a cell-free amplification technique for synthesizing multiple identical copies of any DNA of interest. Okay, next, who invented PCR? Okay, the polymerase chain reaction, or PCR as it is commonly known, was invented by Carey Mullis. Is sorry, invented by Carey Mullis in 1985. Okay, the polymerase chain reaction, it is a commonly known, was invented by Carey Mullis in 1985. Okay. PCR means polymerase chain reaction invented by Carey Mullis in 1985. Next, how does PCR work? PCR, okay, look at here. This is PCR machine, okay? Look at here, okay? Okay. When DNA is copied, replicated prior to cell division, okay, but it is carried out in controlled condition in a laboratory. The machine that is used is simply called a PCR machine or a thermocycler. Okay, test tubes contain the DNA mixture of interest or put into the machine, and the machine changes the temperature to suit each step of the process. Okay, are you understand? This is PCR machine. Okay, next, standard ingredients in the mixtures are the DNA segment of interest. Next, specific primers. Okay, next, heat resistant DNA polymerase enzyme. Next, the 4 different types of DNA nucleotides. Next, the salt needed to create a suitable environment for the enzyme to act. Okay. The DNA segment of interest, okay, means this is the target DNA. Okay. Target DNA means in general, the shorter the sequence of target DNA, the better is the efficiency of PCR. However, in recent years, amplification of DNA fragments up to 10 kilobase pairs has been reported. The sequence of target DNA is also important in PCR. Okay, that is called target DNA segments. Okay, GC. Okay, GC means guanine cytosine. Rich regions of DNA strand in PCR. Okay, next specific primers. Okay, what is mean by primers? Primers play a significant role in determining PCR. Okay, the primers 17 to 30 nucleotides without secondary structure and without complementary among themselves are ideal. The complementary primers can hybridize to form primer dimer and get amplified in PCR. This prevents the multiplication of target DNA. Okay, this is specific primers. Okay, next, heat resistant DNA polymerase enzyme. Okay, DNA polymerase enzyme. DNA polymerase means has already, you know, okay, tag polymerase is preferred as it can withstand high temperature in the hot start protocol. Okay, DNA polymerase is added after the heat denaturation step of the cycle. Okay, next, four different types of DNA nucleotides. Okay, already you know, guanine, cytosine, thymine, adenine. Okay, next, that is four different types of DNA nucleotides. Okay, next, this is enzymes. Okay, suitable environment for the enzymes to act. Next, okay. Okay, this is PCR technique. Okay, first you take DNA strands. Okay, this is DNA strands. First, this is 5 prime, this is 5 prime, this is 3 prime, this is 3 prime. This is phosphate group, this is the hydroxyl group. Okay, OH group. OH means hydroxyl group. P means phosphate group. This is the DNA strands, double helical structure of DNA strands. Okay, you take two strands of DNA. Okay, first step, denaturation step. Okay, what is meant by denaturation? Is a DNA replication. The two strands in the DNA double helix need to be separated. The separation happens by raising the temperature of the mixture, causing the hydrogen bonds between the complementary DNA strands to break. This process is called denaturation. Okay, look at here, don't confuse. First step, denaturation step. You take two DNA strands. Okay, first step, denaturation step. Okay, your understanding purpose, look at here, denaturation step. Look at here, this is the DNA strands. Okay, first adenine binding with the thymine, cytosine binding with the guanine, thymine binding with the adenine. Okay, guanine binding with the cytosine. Okay, this is the DNA strands. Okay, first take DNA strand. Okay, in denaturation step, on raising the temperature to about 95 degrees centigrade for about 1 minute. Okay, the DNA gets denatured and the two strands separated. Look at here. First we take DNA strand. Okay. Next, rising the temperature. Okay. Raising the temperature. Okay. About 1 minute, the DNA gets denatured. Okay. Separated two strands. Look at here. Separated the two strands. Okay. Are you understand? This is the denaturation step. Okay. Look at here. DNA replication, the two strands in the DNA double helix needed to be separated. Sorry. Okay. Are you understand? Next, step two, renaturation or annealing. Okay. Look at here. Okay. First step is done. Okay. Look at here. First, you take DNA strands. Okay. Next, temperature about 80 degrees centigrade or 95 degrees centigrade for about 1 minute. The DNA gets denatured and separated two strands. Look at here. Two strands separated. Okay. Same strands. Okay. Separating. Okay. Next, DNA strands separated. First step is done. Second step, annealing step. Okay. Or renaturation. In this step, as the temperature of the mixture is slowly cooled to about 50 degrees centigrade to 60 degrees centigrade. The primers, look at here, this is primers. Okay. Okay, this is primers. This is primers. This primer base pair with the complementary regions flanking target DNA strands, the process is called renaturation. Look at here. These primers attach with the DNA fragments. Okay, are you understand? This process is called renaturation. Okay, or annealing. Between each DNA strand and the primer rather than the two strands of DNA. Look at here. Add with the primers. This is the DNA strand. Temperature 50 degrees centigrade to 60 degrees centigrade. Okay. Look at here. Annealing step. Primers bind to the target DNA sequences and initiate polymerization. This can only occur once the temperature of the solution has been lowered. Okay. First step, high temperature, it can be maintained. Next step, low temperature. Okay, one primer binds to each strand. Okay, are you understand? Look at here. This is annealing step. This is primers. Okay, look at here. First, this is the DNA strand, same. Okay, step two, and in this step, annealing. Okay, the primers add with the DNA fragments. Okay, look at here. Guanine binding with the cytosine, adenine binding with the thymine, cytosine binding with the guanine, thymine binding with the adenine. Look at here, same. Adenine binding with the thymine, cytosine binding with the guanine, thymine binding with the adenine, guanine binding with the cytosine. Okay, this is the annealing step. Okay, next, look at here. Okay, how do you understand? Primers binds to each strand. Okay, next step, extension. Okay, okay, how do you understand? This is annealing step. Okay, next extension step. Extension or synthesis. Okay, the initiation of DNA synthesis occurs at 3' hydroxyl end. Okay, look at here of each primer. Okay, the primers are extended. Okay, look at here. This is primers. This is primers. These primers. The primers are extended by joining the bases complementary to DNA strands. Okay, the synthesis process in PCR is comparable to the DNA replication of the leading strands. Okay, the temperature has above 70 degrees centigrade. Okay, it can be used Tag polymerase enzyme. Okay, okay, look at here. New strands of DNA are made using the original strands as templates. A DNA polymerase enzyme joins free DNA nucleotides together. Okay, this enzyme is often Tag polymerase, an enzyme originally isolated from thermophilic bacteria called Thermus aquaticus. Okay, the order in which the free nucleotides are added is determined by the sequence of nucleotides in the original template DNA strand. Original DNA strand means template DNA. Okay. The result of one cycle of PCR is two double-stranded sequences of target DNA, each containing one newly made strand and one original strand. Okay, 1 minute. Look at here. This is the extension step. Okay. The initiation DNA synthesis occurs at 3' hydroxyl group. Okay. This is 3' hydroxyl groups. Okay. And of each primer, the primers are, sorry, the primers are extended. Look at here, extended by joining the bases complementary to DNA strand. Okay, complementary strands. The synthetic process in PCR comparable to the DNA replication of the leading strand. Okay, it can be used tag polymerase NJ. Okay, are you understand? The tag DNA polymerase enzyme isolated from the thermophilic bacteria. Okay, Thermus aquaticus. Okay, the tag DNA polymerase is heat resistant, hence it is not necessary to freshly add this enzyme for each cycle of PCR. Okay, are you understand? It can be used tag polymerase enzyme. It is isolated from the thermophilic bacteria. Okay, next. Okay, okay, are you understand? The cycle is repeated many times, usually 20 to 30, as most process using PCR need large quantities of DNA. It only takes 2 to 3 hours to get a billion or many copies. It will become. Okay, are you understand? Okay, okay. It is very simple technique PCR. Okay, don't confuse. Okay, shall I repeat? Okay, look at here. PCR means polymerase chain reaction. What is PCR means? Polymerase. P means polymerase, C means chain, R means reaction. Polymerase chain reaction. Invented by Carey Mullis in 1985. Okay, first. Next. This is PCR instrument. Okay, are you understand? Next. Okay. The polymerase chain reaction is a laboratory technique for generating large quantities of specified DNA is obviously okay. PCR is a self-free amplification technique for synthesizing multiple identical copies, billions of DNA of interest of DNA fragments. Okay, next, next. This is a PCR technique. Okay, ingredients. Okay, first the DNA segments of interest. Okay, this is the target DNA fragments. Okay, target DNA means In general, the shorter the sequence of target DNA, okay, it can be used. Okay, next, specific primers. Okay, primers play a significant role in determining PCR. Okay, primers means 17 to 30 nucleotides. Okay, without secondary structure and without complementary among themselves are ideal. The complementary primers can be hybridized to form prime dimer and get amplified in PCR. Okay, this prevents the multiplication of target DNA. Okay, next, heat resistant DNA polymerase enzyme. Okay, tag polymerase enzyme, it can be used. Okay, next, the four different types of DNA nucleotides means DATP, DGTP, DCTP. Okay, guanine, cytosine, thymine, adenine, DATP. Okay, this is four different types of DNA nucleotide base pairs. It can be used. Next, the salts needed to create a suitable environment for the enzyme to act. Okay, next, total three steps. Okay, first step, denaturation step. First, you take DNA fragments. This is the 5' group. This is 5' group. This is 3'. This is 3'. This is OH group, hydroxyl groups. Okay. Next, this is phosphate group. P means phosphate groups. You take two strands of DNA. First, you take two strands of DNA. Next step, denaturation step. Denaturation means on raising the temperature to about above 80 degrees centigrade for about 1 minute. The DNA gets denatured and two strands separated. Look at here. Okay. First, you take DNA fragment, can apply the heat about 80 degrees centigrade. The DNA strands will be separated. Okay, look at here. Space. Are you understand? Are you observe? Okay. Next, two strands separated. Okay, are you understand? This is the denaturation step. Next, annealing or renaturation. Okay. Okay, look at here. Are you understanding? First step, denaturation. As in DNA replication, the two strands in the DNA double helix need to be separated. The separation happens by raising the temperature of the mixture, causing the hydrogen bonds between the complementary DNA strands to break. This process is called denaturation. Next step, annealing. Okay, look at here. This is annealing step. Okay, this is primers. These primer adding with the DNA strands. Okay, look at here. The primers bind to the target DNA sequences and initiate polymerization. Okay, this can only occur once the temperature of the solution has been lowered. Okay, one primer binds to each strand. Look at here. This is primers. Okay, this primer binding with the DNA strands. Okay, this is the annealing step. As the temperature of the mixture is slowly cooled to about 55 degrees centigrade, the primers base pair with the complementary regions flanking target DNA. This is target DNA. Next, this process is called renaturation or annealing. High concentration of primers. This is high concentration of primers add with the DNA strands. Okay, two primers add with the DNA strands. This is annealing step. Next step, extension. Okay, this is extension step. Okay, look at here. The initiation of DNA synthesis of each primer, the primers are extended by joining the base complementary to DNA strands. Look at here. Okay. DNA strands, the synthetic process in PCR is comparable to the DNA replication of the leading strand. The temperature has to be kept optimal as required by the enzyme DNA polymerase that is called tag polymerase. Okay, this tag polymerase enzyme isolated from the thermophilic bacteria called Thermus aquaticus. The order in which the free nucleotides are added is determined by the sequence of nucleotides in the original DNA strand. Okay, the result of one cycle of PCR is two double strand sequence of target DNA. Okay, look at here. Each containing only newly made strand and one original strand. The cycle is repeated many times, usually 20 to 30, as most process using PCR technique need large quantities of DNA. It only takes 2 to 3 hours to get a billion or copies will become. Okay, are you understand? Total 3 steps. Okay. This is PCR machine. Okay. This picture is okay. Total 3 steps: denaturation, annealing, or renaturation. Next, synthesis or extension. Okay. This picture is from concept understanding. Okay. You should not draw this picture in the examination. Okay. On instead, 1 minute. Look at here. This picture you can draw it. Okay. Or do you understand? Any doubts? This is only steps of PCR. Okay. Any doubts?